Okay, let's talk about something we all rely on, maybe without even thinking about it much. Medical tests.
Absolutely. We get them done. We see the results.
But how do we know they're actually, accurate, trustworthy?
Well, it's definitely not just luck. There's a whole system behind it. Rigorous standards.
And for medical labs, a really key one is ISO 15189, right?
That's the one. ISO 15189.2022. It lays out the requirements for quality and competence.
Got it. And for this deep dive, we're actually looking at excerpts from a Simplified Handbook on this standard.
Yeah.
Kind of breaking it down.
Exactly. We're using that material as a guide, sort of a mini tutorial, to unpack what quality and competence really mean in a medical lab. That's the mission here.
Perfect. So let's get into it. What really happens behind the scenes to make those test results reliable?
Okay, so the handbook starts by defining the scope. What ISO 15189.2022 is actually for.
Right.
And it clearly states it outlines the specific requirements for quality and competence, but specifically in medical laboratories.
Okay, so labs handling human samples, providing results for, diagnosis or treatment.
Precisely. And it's primarily for the labs themselves, first off, to develop their own quality management systems and assess their own competence.
But it's not just for them, is it? I think the handbook mentioned others use it too.
Oh, yeah, absolutely. It's crucial for lab users, think clinicians, hospitals.
Maybe even us, patients?
Potentially, yes. And definitely for regulatory authorities and the accreditation bodies. They use this standard to confirm that a lab is competent. Getting accredited means they've passed an independent check against these requirements.
That makes sense. And the handbook highlights something interesting. It also applies to POCT, point of care testing.
Yes, that's important. POCT, point of care testing, is any testing done near or actually at the patient's side?
Like those rapid strep tests you get right in the doctor's office.
Exactly that. Or blood glucose meters used at the bedside in a hospital. Even some home tests could fall under this umbrella depending on the context. It's testing outside the central lab.
And the standard still applies.
The requirements still apply. That's a key takeaway from the source.
Okay. And there was a note, too, about other rules.
Right. The handbook points out that while ISO 15189 provides this core framework for quality and competence, labs also have to follow any other specific regulations, international, national, or regional, that might cover the same ground or add more requirements.
So it fits into a bigger picture of rules.
Definitely.
Okay, that sets the scene. Now, standards like this, they don't just appear out of nowhere, right? They build on other work. The handbook calls these normative references.
They absolutely do. They integrate requirements from other foundational standards. Our source highlights three main ones.
First up is ISO IE Ski Guide 99.2000, VIM.
VIM, yes. The International Vocabulary of Metrology.
Metrology, the science of measurement.
Exactly. And what's fascinating, like you said, is that before you can standardize the tests, you need to standardize the language of measurement itself. Everyone has to mean the same thing by basic terms.
Makes sense. And ISO and IEC are the big international bodies doing this standardization.
Correct. International Organization for Standardization and the International Electrotechnical Commission. Global players.
So shared language for measurement is step one. What's reference number two?
That's ISO IEC 17000.2020. This one covers conformity assessment. Basically, the vocabulary and principles for checking if something, a product, a process, like our medical lab, actually meets the requirements set out for it.
Okay, so how do you check compliance? And the third one connects to labs more generally.
Yes, that's ISO IEC 17025.2017. This is the big one for general requirements for the competence of all kinds of testing and calibration laboratories.
So 500-189 takes the principles from 17025.
Right, and tailors them specifically for the medical lab setting with all its unique needs and impacts on patient care. But it's built on that general lab competence foundation.
And the handbook mentions a rule about dates for these references.
Yeah, it's pretty strict. If a standard is referenced with a date, you have to use that specific version. No wiggle room.
And if there's no date?
Then you use the latest edition, including any updates or amendments. It just ensures everyone's on the same page about which version of the building blocks they're using.
Clarity is key, I guess.
Essential for the whole system to work.
Okay, so we've got the scope, the foundations.
Yeah.
Now let's dive into the language itself. Section 3 of the standard, according to the handbook, is all about terms and definitions.
And this is crucial. You really need to understand the vocabulary to apply the standard properly. It's not just, academic.
Right.
The standard uses terms defined in VIM and 17,000, as we mentioned, but it also adds its own specific definitions for the medical lab context. The handbook even points out you can find these online on the ISO and IEC platforms, like living dictionaries.
Handy. So let's walk through some key ones the handbook highlights. First up, bias or measurement bias.
Okay, bias. The handbook defines it as an estimate of a systematic error.
Systematic meaning, not random.
Exactly. It's an error that consistently pushes results in one direction, away from the true value.
Like the example they use.
Yeah.
The thermometer.
Yeah, the classic example. A thermometer that always reads 1 degree Celsius too high. That consistent +1 degree, that's the bias.
And you need to know about it, obviously, to correct for it or account for it.
Absolutely. Uncorrected bias can lead to wrong interpretations.
Makes total sense. Okay, next, biological reference interval. This sounds familiar, like something you see on your results printout.
It is, often, yes. The definition is a specific range of values for a test, and it's derived from what they call a biological reference population.
So like a group of healthy people.
Typically, yes. A defined group, usually healthy. This range then gives doctors a baseline to compare your result against.
This is what people often call the normal range, isn't it?
It is, but the handbook actually cautions against using vague terms like normal range. Biological reference interval is the precise term.
Why the precision?
Because what's really fascinating, and the handbook stresses this, is how specific this interval is. It depends not just on the population, but on the exact type of sample used and the specific testing procedure the lab employed.
Wow, okay. So my lab's reference interval might be slightly different from another lab's if they use a different method.
It could be, yes. The handbook also notes it's usually the middle 95% of results from that reference group, but sometimes it might be asymmetric or just an upper limit or just a lower limit, depending on what makes clinical sense for that test.
That level of detail matters for interpretation. How's that different from a clinical decision limit?
Good question. A clinical decision limit is different. It's not based on a healthy population range. It's a specific value for a test result that signals something important, like a higher risk of a particular disease, or a level that helps confirm a diagnosis or guides treatment.
So it's like a threshold for action.
Exactly, a threshold value.
Yeah.
The handbook gives the example of cholesterol, say, a level over 200 mg/dL might be linked to increased heart disease risk. That 200 number acts as a clinical decision limit.
Okay.
And the handbook also mentions that for monitoring drug levels, these limits are often called the therapeutic range, keeping the drug within that window for effectiveness without toxicity.
Got it. Okay, now the handbook flags one that sounds, well, complex. Commutability of a reference material.
Oh yeah, this one's really interesting and fundamentally important, but definitely a bit technical.
We'll try to break it down.
Okay, so the handbook defines commutability as a property of a reference material. Think of things like calibrators or control samples that labs use.
The stuff they use to check their machines are working right.
Exactly. Commutability means that this reference material, it behaves, it reacts in the same way that actual patient samples do when you test them using different methods or different instruments.
Behaves the same way.
Mm-hmm.
Why is that so critical?
Well, think about it. You want results to be comparable, right? If a patient sample gets tested in Lab A on machine X, and then maybe later in Lab B on machine Y.
You want the results to mean the same thing, ideally be the same number if the patient hasn't changed.
Precisely. For that to happen reliably, the calibrators and controls used on machine X and machine Y need to interact with the test systems in a way that mirrors how real patient blood or urine or whatever interacts if they don't.
Then calibrating machine X might give a slightly different meaning compared to calibrating machine Y, even if they use the same value calibrator.
You've got it. If the calibrator isn't commutable, it might react differently to nuances in method X versus method Y compared to how a patient sample reacts. This non-commutability can introduce discrepancies between methods.
Wow. So commutability helps ensure results are truly equivalent, or at least comparable, across different platforms. That's huge for consistency in healthcare.
It's massive. The handbook adds some notes on this, how it's usually checked by testing the reference material alongside a bunch of patient samples on the different methods you care about.
Comparing the patterns.
Right. And comparing across multiple methods gives you a better picture. It's about fitness for purpose, too. How much agreement you need depends on the clinical situation. And critically, a commutability statement is specific. It only applies to the material and the exact methods it was tested with.
It's not a universal property, though.
No, it's very context dependent, but incredibly important for reliable, comparable results across the board.
Okay, that's a deep one, but vital. Let's shift to the human element. Competence.
Competence. The handbook definition is pretty clear. The demonstrated ability to apply knowledge and skills to achieve the intended results.
So basically, proving you know your stuff and can do the job right.
That's a good way to put it, yeah. Being good at what you do in the lab. The example used is a technician who can accurately analyze samples because they have the right training, they follow the procedure correctly, they have the skills.
It's demonstrated ability, you have to show it.
Exactly, not just theoretical knowledge.
And what happens when things don't go right? That leads to the next term, complaint.
Right. A complaint, according to the handbook, is an expression of dissatisfaction. It comes from a person or an organization directed at the lab.
And crucially,
a response or resolution is expected, either explicitly asked for or implicitly understood.
So it's not just venting. It's flagging an issue with the expectation the lab will look into it.
Precisely. The example is a doctor filing a complaint because results were maybe wrong or way too slow, and it affected how they treated a patient.
Got it. How about consultant? That seems straightforward.
It is. A person who provides professional expert advice.
Like bringing in an outside expert.
Yep. The example given is hiring a quality management consultant to help the lab improve its processes to meet ISO 51589 requirements. Someone with specialized expertise.
Okay. Now let's look at the word examination.
In the world of this standard, examination means the actual set of operations done to figure out the value or characteristic of something in a sample.
So it's the official word for lab test.
Pretty much, yes. That's the formal term the standard uses. The handbook notes it can involve many steps, and the result can be a number quantitative, like blood sugar level.
Or just a yes-no type answer.
Right. Qualitative, like whether a certain bacteria is present or not.
Yeah.
And yeah, people often use assay or just test interchangeably in practice.
And flowing from that, examination procedure.
This is the detailed, documented, step-by-step instruction manual for how to perform a specific examination. It's based on a chosen method.
The specific recipe the lab tech follows every single time for that test.
Perfect analogy. That's exactly it. The handbook emphasizes its purpose is consistency and accuracy. Make sure everyone does it the same way every time.
I bet that's critical.
Absolutely. The handbook mentions it's used heavily with IVD medical devices in the kits and instruments.
In vitro diagnostics tests done on samples outside the body.
Right. And labs using those devices rely heavily on these detailed procedures. And again, you might hear terms like analytical method or test procedure used to mean the same thing.
Okay, so we've walked through quite a few definitions here. Bias, reference intervals, decision limits, that complex but crucial commutability, competence, complaints, consultants, and the terms for tests and procedures. Stepping back, how do all these definitions, taken together, really build that foundation for trust in the results you get back?
Well, what really stands out is the sheer level of detail and control required. It's not just about having, say, a good machine or smart people.
Right.
You need this incredibly precise, shared language for everything. What counts as an error? Like bias? What does normal actually mean? That's the biological reference interval. When do we need to act to the clinical decision limit? How do we make sure results from different places compare commutability?
And proof that people can do the job competent.
Exactly. And procedures for handling issues like complaints. Each definition we discussed, it represents a specific control point, a layer of the quality system designed to make that final result reliable.
It really drives home that a single number on your report isn't simple at all. It's the end point of this very structured, very standardized process.
Absolutely. It's the result of this kind of invisible infrastructure that underpins reliable diagnostic testing.
So let's wrap up this deep dive, drawing on those handbook excerpts on ISO 45189.2022. We looked at the standard scope, who it's for, including point-of-care testing.
We saw how it builds on other key standards for measurement language and general lab competence.
And we spent a good chunk of time unpacking some really vital terms from Section 3 definitions like bias, biological reference intervals, clinical decision limits,
That really critical concept of commutability.
competence, complaints, consultants, and the formal terms,
Yeah.
examination and examination procedure.
And hopefully understanding these terms and requirements gives you, the listener, a much clearer sense of the rigorous system that's in place behind the scenes in accredited medical labs.
It definitely shows the layers involved. Which leads to a final thought. Think about how much precision goes into just the terminology. Using biological reference interval instead of normal range, defining biases so specifically. It's all aimed at clarity and reducing error. What might be the real-world consequences for patient care if that level of semantic rigor, that demand for precise language, wasn't embedded in these standards? Something to consider.